back, Phelan-McDermid syndrome community. I am honored today to have Dr. Peça, who is the 2024 PMS Grant Awardee for the Translational Award. He is an assistant professor from the University of Coimbra in Portugal, and he will be talking about his research today. Welcome.
Well, thank you, Lauren, and it's a pleasure to be here and to share some of the science and some of the work that we're doing with support from the PMS Foundation. Thank you.
Great. And before we get kind of into the updates about your specific project, which is called the non-canonical role of SHANK3 in early development, can you share with our families a little bit about your background and how you found your way into Phelan-McDermid syndrome research?
Yeah, sure. So, I was growing up in Portugal. I can start by saying that, and I did my undergraduate degree here. And then, as soon as I finished, I was given the opportunity to get on a fellowship that would allow me to select a lab to perform my PhD. So this was a great opportunity because basically I was given this possibility to decide where to go to pursue my research studies, my doctoral degree. And so I ended up joining the lab of Guoping Feng at the time at Duke University. And I started my foray into the research in neurobiology, originally doing some work in the field of optogenetics. And then also as Guoping's lab was transitioning to MIT, I decided to keep going with some of the work that we were doing, and I decided to invest quite a lot of time on generating, let's say, new animal models that would allow us to understand this gene that we were interested in, which was called SHANK3. And at the time, we didn't really understand precisely that SHANK3 was at the core of Phelan-McDermid syndrome as we know today. So we kind of started looking at it from a very fundamental aspect, just curiosity about the role and the function of this gene and the protein that it coded. But then it became very obvious very soon that we were working on something that was actually quite interesting, quite novel, and quite relevant. And so I would say that this was the start of my interest in this disorder. It started basically as a pure, let's say, biological curiosity, and then as we went deeper and deeper, we started understanding this deep connection with a very important human disorder.
Wow. Yeah, that's really impressive and sort of such a, sort of fascinating tale, kind of including the history of Phelan-McDermid syndrome that you sort of stumbled upon SHANK3 in your work and turns out it becomes, it's a really important and, devastating consequence that leads to Phelan-McDermid syndrome. So that it's kind of cool to start at the kind of ground floor of this and then being involved, it sounds like, in the generation of sort of these mouse models, which are so essential to our translational science.
Yeah, no, exactly. So to give you a little bit more of a historical context, so the field, the area that we were really interested in understanding a little bit more is a region of neurons called the postsynaptic density, which is an area at the synapse where neurons use to communicate with one another. And at the time when I was starting my PhD, one of the hot topics in the field, let's say, was really trying to understand what was the function of the proteins that were making up this sort of postsynaptic density. And the reason it was called density is because it was really packed with a lot of proteins, making it a structure that is very dense, as the name itself says. And so SHANK3, SAPAP3, PST95, just a few names of proteins that we knew were highly, highly enriched in this structure, but whose function we actually knew very little about. And so we were in the Duke and several other places were working on understanding what were the specific function of some of these proteins and understanding the biology behind them, and really a clear understanding that if you mutated or if you damage the expression of these proteins in some ways, even in very simple animals like mice, you would see fantastic changes in the behavior of these animals. And so that immediately told us that we were looking at the structure and genes and proteins that were quite important. And then as data from human genetics started to becoming, providing more and more solid evidence for this, it became clear that there was a link between some of the genes that we were interested in looking at and their consequences for human health.
Congrats again on winning this award, and we're really excited for your work here. And so, for our families listening, could you tell us briefly what your project aims to accomplish?
Of course, one thing that is particularly curious in the case of PMS and the models of, let's say, SHANK3, um, is that we detect quite a lot of changes in synaptic and neuronal activity. But some of these changes, like the real root cause, is not well understood. And so if you're a curious person, if you're a curious individual, you can look at it fundamentally in two ways, right? You want to treat, cure, and try to mitigate, let's say, some of these changes to hope that you can help, like you can create the novel therapies, drugs, and the treatments. But there's always some interest in also trying to understand where are these changes actually coming from. And so this the project that we're doing right now is really focused on both of these aspects. Actually, we're lucky again in the possibility to work on both aspects at the same time. An aspect that is more of a biological curiosity, but at the same time, it is opening up a new avenue that could potentially help us mitigate some of the changes that occur in the brain when there are mutations in this very important gene. So to be a little bit more specific or a little bit more technical, if I may, what is, let's say, the canonical role of SHANK3, as I was saying before, is as a very important protein in this postsynaptic density. We often say that SHANK3 is a master scaffolding protein, so helping organize this structure and helping neurons communicate with one another. However, what we have found now, and there have been some evidence in the literature that haven't, maybe no one has paid too much attention to them, but it really piqued our curiosity, is that SHANK3 also seems to be present in the nucleus of certain cells. And now we found that SHANK3 is really enriched in the nucleus of neural progenitors. So these cells that give rise to both neurons and other glial cells in the brain. And so the question then is, why would you have this critical synaptic protein being present very early on in the nucleus? Is it playing a role there? Because then if it is, that could mean that SHANK3 is already playing a role even before neurons themselves start having synapses and communicating. And that could be quite relevant in order for us to understand disease progression and some of the alterations that actually then show up. Another aspect that we want to explore is that we think that we have an idea of the partners of SHANK in the nucleus. And again, these haven't really been explored. And so this could be something that could be worthwhile looking at. And this is what we proposed to do, and for which you very generously gave us some funding now to explore in greater detail.
Yeah. It's fascinating. And, my background's in clinical psychology. And while I dabbled in some molecular phenotyping work basic science is not necessarily, did bio 101 and some anatomy and physiology in college, but I do find it so fascinating in that, you are or have identified that SHANK3 is in the nucleus of sort of these like early precursor cells that, I don't want to extrapolate or we don't know enough, but as you point out, for them to be there, it doesn't seem completely random. There's got to be some reason. And to think that there could be some reason SHANK3 is there and could help determine, the development of glial cells or neurons or whatever, seems to suggest that SHANK3, has more than just one really important role. And sort of identifying that better and then figuring out kind of the other genes or partners associated with it could really lead to some impactful findings, and expand maybe some treatment targets. So, where are you with the project now? Any kind of new results you can tease us with?
Yeah, so there are a couple of things that we've already been able to figure out that are quite exciting. But as you well know, research projects take their time to mature, and so we knew that SHANK3 was in the nucleus, but we didn't know where. So we could see that there was some specific localization. I'll be a little bit technical in this part, but the nucleus is actually a rather complex structure. We tend to think of the nucleus in the cell as this, let's say, compartment for DNA, but it's actually far more than that. There's sublocalizations within the nucleus. And what piqued my interest was that, and we put this in the proposal, was to identify specifically where SHANK3 is, because we were saying it's in the nucleus, and that is crystal clear, but the enrichment pattern, so meaning that we were seeing some areas inside of the nucleus that showed much higher, much stronger signal. That leads us to speculate that potentially SHANK3 would be enriched in subnuclear compartments, so meaning that within the nucleus, it could be localizing into specific structures. And these structures can have a variety of natures and functions. And one of the exciting updates that we have is that we're now able to state or say that SHANK3 seems to enrich in the nucleolus. And so the nucleolus plays quite a lot of interesting roles, is involved also in the regulation of ribosomes, and has specific functions that are something that don't have just to do with packaging or compartmentalization of DNA. And that already gives us some ideas or validation in terms of the partners that we expect SHANK3 to interact with. So that's important because of that. We want to make sure that we are able to see where SHANK3 is, and then also see if these partners that we expect to be interesting or relevant to also be present in the same location. So it gives us a place where to look more intensely. Another aspect that we were able to already get some interesting results is on this very powerful technology that is now, being used quite widespreadly, I would say, in the field of biomedical research, which is called single-cell RNA sequencing, which is a technique that allows us to see what, in a complex tissue, what each individual cell is actually expressing. So, whereas if you have a complex, let's say, tissue again, all the cells will have the same information in their DNA, all the same, components will be present there. But what each particular cell is expressing is unique. And so this is a technology that allows us to see what each individual cell is doing. And we're using it to basically compare the expression profiles in human brain organoids, which are these models, these 3D models, that we start off with let's say, control cell line and the cell line that has a mutation in the SHANK3 gene. Okay, and so we create these 3D cultures. We let them grow for a couple of weeks or a couple of months, and then we use this single cell RNA sequencing technology to see what is actually going on. The reason we decided to do this, and this may be, let's say, give you a more clearer picture of what we're trying to do, is again this idea of SHANK3 is in the nucleus of the precursors. It could be affecting how these precursors give rise to neurons or give rise to glia, or even the balance of the number of cells that get generated. And so the single cell RNA sequencing gives us a lot of information, but it can also be used as a sort of census, telling us, what are the numbers of different types of brain cell populations that are coming up when you're in control conditions or when you have the mutation. So I cannot tell you precisely where the changes are, but what I can tell you for sure is that there are differences in the ratios of the different cell types that are showing up within these models, which again, we find quite exciting.
Great. So for our listeners, you're first sort of identifying, where is SHANK3 in the nucleus? You wanted to figure out sort of, you could find it and locate it. And that's going to tell us a little bit more potentially about the role, but then as you mentioned, sort of its specific partners, which also could be implicated in Phelan-McDermid syndrome. And so far, it looks like it's in the nucleolus, which is a new finding, to the best of my knowledge. And so that's exciting and gives us some specific information. And then this other part is sort of using this fancy technology to really, I like your analogy of a census, of really figuring out what is that ratio of glia to neurons. My follow-up question is depending on the results, what is that going to tell us? Like if you find out that there's more glia versus neurons or the other way around, what does that tell us about the mechanism?
That's a great question. So one of the contributions that I gave to the field and obviously Guoping gave to the field in terms of characterizing the consequence of alterations in the SHANK3 gene was that we found that there's a perturbation in the corticostriatal pathway. So it's projections that come from the cortex down to the striatum. And so we are seeing alterations in communications in this specific brain circuit. There are many other contributions, of course. I just want to highlight maybe 2 or 3 aspects that will be important to understand the answer to the question that you just posed. So this circuit is specifically affected. We know that ultimately the cause is the SHANK3 mutation, but we don't know how we're getting to this specific problem in communication. There was then another contribution by a set of colleagues that came out of Harvard, that they showed actually that yes, indeed, this alteration in corticostriatal circuits is there. And they added to this knowledge by figuring out and seeing that there's actually a hyper excitability in the cortex that occurs early on. These works were done in animal models. The story becomes even, I would say, more interesting when another colleague in Stanford recently showed that when you go back to these human brain organoids, these 3D cultures that we get in the lab, they also see some evidence of hyper-excitability very early on again. And so the story can kind of, in the field, is kind of like at this point where we know that there are alterations in activity. They seem to emerge at least early on in the cortex, but we don't know how it actually starts. Now, going back to the question that you asked, why is it important to know if the census of cells is different between control and in the presence of a mutation, SHANK3? Well, because if we see, for example, that there's an acceleration in the appearance of, let's say, excitatory neurons, or if there's a delay in the appearance of some type of inhibitory neurons in the cortex, that could actually be another, let's say, breadcrumb in the
Yeah.
trail that we have to find in order to say, okay, this is the earliest point that we can find in terms of being able to, let's say, alter the trajectory of where the disorder is emerging from. So this
Mhm.
is critical, let's say, from a fundamental biology aspect. This is, again, following the breadcrumbs, trying to understand where the initial insults are coming from.
Right. A nice segue into this broader discussion around translational science and really trying to take these breadcrumbs that you're suggesting and move them down the line so that they can be actionable and turn into human clinical or therapeutics.
We know something about animal studies, and we
Hmm.
know that if you re-express SHANK3, that is sufficient to get quite a lot of improvements. And so the question then becomes, if you can get that type of outcome in an animal model, really, what is the impact that you can get in a human? So the idea that by introducing expression of SHANK3 in a human could have quite important outcomes, right? We shouldn't forget, though, that there is some developmental trajectory that you're not going to roll back in time, right? There are critical windows, there are critical periods of development, and that you miss, right? And um, were already impacted. That may be harder to, but um, to completely reverse. But there's this hope for sure that it's something that we could act upon, right? What I'm trying to suggest is that by really understanding if this neurodevelopmental aspect of SHANK3, if it turns out to be really important, that may also help us guide future therapies. So that may help us understand how early we actually could potentially need to go on one end. On the other end, it could mean that some of the targets that you need to hit in the nucleus is something that you need to be paying attention to, as
Mhm.
opposed to just the synaptic role
Mhm.
SHANK3, right? And
Right.
so the strategies that are now being designed and where we're putting quite a lot of hope that they work are really focused in the synapse. So for sure that that role is going to be covered. But if it turns out that SHANK3 is also important in the nucleus, we need to know that, and in order to evaluate and to iterate on future strategies.
You do bring up a really good point because, I think this is of interest to a lot of families is as these various gene RNA therapies are coming online and thinking about when and what age are being used. And so, I do appreciate you sort of pointing out that it's regardless of whether it's a little bit older or a little bit younger, these are still going to be impactful. And it's just sort of, you know, we'll have to wait and see sort of what that impact looks like. And what I'm hearing is a lot of new potential avenues for therapeutic development, whether it's, as you point out, going inside the nucleus and figuring out intervention we can do there. And then also this idea of sort of this early developmental period too that you're identifying, that maybe we need to correct the inhibitory or maybe we need to correct the excitatory circuits. And so your work is laying some really important foundation for what the potential or future of therapeutics in Phelan-McDermid syndrome could look like.
Yeah. So again, the part of our focus right now with this grant is better understand the interaction partners of SHANK3 in the nucleus and in the nucleolus. So basically characterize this network of interactors around it. So if I had to lay out our hypothesis, is that in the nucleolus, SHANK3 could be by being present there, and also thinking of its role as a scaffolding protein, it could be kind of like, let's say, capturing specific proteins, trapping them in the nucleolus, and pulling them away from the nucleus. And so if you think about this type of, let's say, sequestering capability of SHANK3, or this putative sequestering capability of SHANK3, I should say, you could envision a role for this protein to actually regulate gene expression. And if you're regulating gene expression this early on, this is again what would then trickle down into changes into how early you start seeing neurons.
Right.
How many cells of type A, type B, or type C are developing, and what are the time points at which they are showing up.
And so, you know, as you're starting to get some findings and thinking about next steps, I mean, is there a particular direction or next sort of grant that you've already started writing in your head to answer sort of the next question?
We have a target already that now we first we need to validate it, of course. But what is interesting is that this is a druggable target. So meaning that it's a target for which that is already actually associated with neurodevelopmental disorder with
Okay.
characteristics similar to Phelan-McDermid. But at the same time, it's a protein that is uh, being studied in the field of cancer.
Okay.
And there's a compound, a small compound that has been developed as a target for this partner, and so now we want to test if introducing this compound by perturbing the activity of this partner of SHANK, or this putative partner of SHANK3, I should say, can we actually um, perform changes that go in line with what we expect. So on one hand, we have this alteration in Shank that is, we theorize, altering the activity of this partner. And so we're now asking the question, if if I use this small compound that is already available, can I actually curb how
Mm.
neurodevelopment is occurring? So this is the, very ambitious down the line project. But I should say that having this, you'll excuse me for saying this, but a smaller grant like the one that you've awarded us is really critical, because it allows us to start to get, for example, the animal approvals and authorization to start some very small scale studies that then will give us the ability to generate preliminary data to go for much more ambitious projects. So it's quite exciting.
Yeah, it is exciting. And I really appreciate you pointing out the importance of these sort of seed grants, for lack of a better term, that really provides enough money, maybe, to start the projects going and really generating that preliminary data that's going to be used and is essential for those bigger grants. We're never going to claim that we're going to be an RO1 or the equivalent in Portugal size grant, but we really want to make sure that we are providing funding to researchers to get some new project, new idea off the ground so you can get that. And I think this is a great example of how one small project can lead into so many different directions. And I love to hear that you have students, trainees in your lab that could be the future of Phelan-McDermid syndrome research. And so getting them involved and excited so early is going to be really important. And I know, sort of kind of as a segue, you mentioned that you had one of your trainees go to last conference, PMSF family conference. And so, I think that's so important in this basic research translational field, that these researchers, young trainees, have access and interactions with our community to really put a face to the name that they're studying. So, are you hoping to be able to get to our next one in 2026?
Yes, absolutely. I'm hoping to be able to go to the US and show some of the updates that we have in person, and talk to you, talk to the families, and also some of the other colleagues working in the field. And just like you're saying, these grants are really important to kickstart and kickstart projects and get some ideas flowing. And definitely having a young researcher, in this case a PhD student, already being trained in the field really guarantees that the ideas keep going in the future. You always have researchers popping in and out of a specific field, but definitely in this case we're already making sure that the story can keep going in the future.
Absolutely. So we do need to wrap up, but before we go, I did want you to share the fun fact about the University of Coimbra that you shared with me before we started recording, because I think it's something our community would enjoy.
Of course, yes. So the University of Coimbra is actually one of the oldest universities in the world in continuous operation. It's a UNESCO historical site, so it's attracting quite a lot of people just for the historical reasons and being able to visit such an important place in the history of Portugal. Coimbra was actually the first capital of Portugal, and the university remained as, let's say, the educational capital for quite a long time in our country's shared collective culture. And regardless of being old university, the research centers are actually quite fresh.
It's not from the 13th century?
Not from the 13th century. The research institutions are much younger, much vibrant. So
Yeah.
I invite everyone to come. Last year we just had the first meeting of the Portuguese Phelan-McDermid Syndrome Association. It was a huge success. We had experts from all over the world coming, and it was really focused on the families, but also really focused on the science. And it was a pleasure hosting everyone here in Coimbra, and we hope to do it again. Our colleagues, our neighbors next door, are now organizing in Spain a meeting this year. So I also like to say that I'm sure that it will be a huge success and a great opportunity to share some information. Unfortunately, I will not be able to go due to calendar issues, but I'll see you in 2026, I'm sure.
Yes, excited to see you in 2026, and maybe we can convince the Portugal group to get another conference going and get there in the next year or two. Well, thank you so much, Dr. Peça. Really appreciate this time and really informative and insightful kind of not only history of Shank 3, but also kind of what you've been doing. Surely we'll keep updated on your work and excited to see where it goes. So thank you and congrats again on getting this 2024 PMSF Translational Award. And again, I always want to thank our donors who really make these grants possible for the community. And as you sort of heard here, it's really foundational for future research, but also sort of building the next generation of Phelan-McDermid researchers. So thanks again.
Thank you for the support. Thank you.
Of course. All right, tune in out and stay tuned for next episode in a couple weeks. Thanks so much.